Section 8.7, Mutations. Mutations are changes in DNA that may or may not affect the phenotype of the organism. There are different types of mutations, those that affect a single gene and those that affect an entire chromosome. If a mutation is happening during replication, it's going to affect a single gene. And mutations that occur during meiosis affect a group of genes or an entire chromosome. A point mutation is a type of mutation where one nucleotide is substituted for another. The correct nucleotide is replaced with an incorrect one. So most of the time, DNA polymerase, when it edits and corrects this, will fix this kind of mistake. If it doesn't, because there's always that chance that it doesn't, it can affect the entire protein. There are three different types of gene mutations called substitutions. And the first, a missense mutation, it causes a change in the amino acid that's coded for by the triplet on DNA, which makes the codon on RNA, which is translated into the protein during translation. For example, if the correct codon is UUU, which codes for phenylalanine, and because of mutation, that becomes a UUA, which codes for leucine. We're getting a different amino acid than you should have at that spot in the protein, and it's going to change the structure a little bit. Silent mutation is another type of substitution where, in this case, GUU, which codes for valine, and the mutation causes the codon to become GUA, which also codes for valine. So even though there's a change in the base, the amino acid sequence doesn't change, so it doesn't affect the protein. And the last one of these is a nonsense mutation, where the amino acid is changed to a stop codon. Okay, in this case, UGU, which codes for cysteine, gets changed to UGA, which is a stop codon. And what that does is it just stops translation at that point, and so the protein is never fully made. It could be missing a major part of it or enough of it where it's going to affect its structure, making it unfunctional. Okay, so here's a couple of examples of these substitution mutations. In this first one here, okay, the original code is ATG, GAA, GCA, and CGT. That would be the DNA code. You can tell it's DNA because there's no uracils there. ATG would code for methionine. GAA would give the codon that would give you glutamine. This will give you alanine, and this will give you glycine. In a mutation, this just one base, this substitution, A, becomes G, and what it does is it still codes for glutamine. So this would be a silent mutation, because although there is a different code on the DNA, which causes a different messenger RNA transcript to be made, which causes a different, or it causes translation to occur, it doesn't affect the amino acid sequence, so it is silent. Okay, in this case, what's happening is there's a mutation here, G goes to T, which is going to, rather than getting glutamine, it's going to give a stop codon. When a stop codon is coded or is produced instead of the amino acid, this is a nonsense mutation. And last, on this one, the mutation here, A, gets replaced with a C, and what happens is rather than getting glutamine, this one, we're getting aspartame, which is a different amino acid. The rest of the chain remains the same, but because this is a different amino acid, it's going to tweak the structure of the protein enough where it would be non-functional. And this is a missense mutation. A frame shift mutation involves insertion, meaning adding, or deletion, meaning subtraction, of one or more nucleotides in the DNA sequence. What this does is it changes the reading frame for the codons that are being read on messenger RNA, and it's going to affect the protein more than just a point mutation because it's going to shift everything over for the rest of the chain, which can cause major differences in how this is getting read. So in a deletion mutation, like here, remember this is removing one or more nucleotides from DNA, so that would affect the RNA, messenger RNA. So this is our messenger RNA here. Okay, and what we're showing here is that this is what would be normally coded for by this messenger RNA transcript during translation. So we would get the methionine, leucine, glutamine, valine, arginine here, and just by removing this U, what it does to this reading frame is we still get AUG, but for the second one, rather than getting CUA, we're getting CAG, because CAG, see how this is shifting over one? And we get AAG, UCCGA. Everything from this point on is going to change the amino acid sequence coded for by the messenger RNA. So that could be a major change to the protein. Okay, and insertion does much the same thing, but in the other direction. So again, this is our messenger RNA transcript, and in this case, we're going to add in a G right here. So this would be adding in a G at this point on the original. And again, what it does is the first two, no problem. They're going to be the same. But after this, it's going to change from glutamine to glycine. And then valine would be serine, arginine would be proline. And it's changing everything again from after that point because the reading frame shifts. You're adding an extra base, so the 3 bases in the codon would be moved over 1. If we inserted 2 nucleotides, then the reading frame would move over 2. So it would affect the amino acids coded for after that point. Okay, so in this one, what is happening is we're getting a mutation. Remember this is the original, and this is after the mutation. Here, everything's the same, everything's the same, but rather than UAC coding for tyrosine, there is a point substitution, a point mutation, this substitution, where C gets replaced with G, and rather than coding for the amino acid tyrosine, it codes for a stop codon. Remember, when a stop codon replaces an amino acid, this is a nonsense mutation. Okay, in this case, what's happening is we're getting a different type of point mutation. This time, what's happening is there's an insertion. We're getting an extra base added. Okay, so same. But here, what's happening is we're getting this uracil inserted here between the U and the A. And what it does is we get a frame shift mutation. So everything is moving just a little bit. Rather than coding for UAC, it's going to code for UUU. This becomes CGG, and then we have this extra little base here. So again, this is an insertion. Okay, and it's a type of point mutation. Okay, another type of mutation is chromosomal mutations. Now, chromosomal mutations happen during formation of gametes, which happens during meiosis. And during meiosis, crossing over can occur during prophase 1 to meiosis if the segment that was crossed over or should have been crossed over reattached incorrectly, a chromosomal mutation will occur in its chromosome because it's affecting more of the chromosome, like a bigger chunk of the chromosome. One of these chromosomal mutations is called a gene duplication. If both segments of the homologous chromosome, or homologous gene, I should say, reattach to the same chromosome, what you're getting is an extra gene or a duplicated gene on one of those chromosomes, and it's missing on the other. So this would be a gene duplication on the one that gets the extra and a chromosomal deletion on the other. Another type is an inversion. Inversion just means to flip, okay? So the segment that is supposed to attach to the homologous chromosome, it does attach, but it attaches in inverse order. It flips, so it's going a different direction. Another type of these chromosomal mutations is a translocation. A piece of one chromosome moves to a different non-homologous chromosome and attaches there, which is, again, creating a chromosomal mutation. And the last type of these chromosomal mutations would be non-disjunction. This would be where the sister chromatids don't separate during meiosis, and you're going to get polyploid. You'll get an extra chromosome. This would be when all chromosomes fail to separate. This happens in plants. In humans, if chromosome 21 doesn't separate in the egg and that egg gets fertilized, you'll get trisomy 21, which causes Down syndrome from this non-disjunction or chromosomal mutation. So here are some examples of these chromosomal mutations. A deletion. Okay, deletion, what happens is this gene ends up missing. Okay, it's just lost out of it. A duplication, where these two genes, BC, are duplicated. They both go to the same chromosome, so we get an extra gene duplication. An inversion, where BCD attaches, but in a different order. It goes to DCB, so this means inversion, they're flipped. Okay, reciprocal translocation, where these guys, AB and MNO, which are non-homologous chromosomes, go to the non-homologous chromosome and attach there. So you're getting a shuffling of the genes, but it is a chromosomal mutation because it's not supposed to work that way. Mutations may or may not affect the phenotype of the organism. The effect of mutation depends on how many genes are involved and the location of the mutation on the gene. Okay, so these chromosomal mutations impact on phenotype. Chromosomal mutations can affect a lot of genes, and as a result can have a large effect on the organism. It can cause a gene to no longer function, or it can cause a new variation of a gene. Gene mutations occur on a smaller scale, but can still have a big impact on the phenotype. Many of them, however, don't affect the phenotype at all. Mutations can happen in both body and germ cells. Remember, body cells are somatic cells. Mutations in body cells only affect the organism. If they're in your body, they're not going to get passed on to your offspring, so they're only going to affect the person who has the mutation. Mutations in germ cells, since these are the sex cells, will be passed off to offspring, and as a result, they are a source of genetic variation, and it's going to affect the phenotype of the offspring. It can either be positive or negative. Mutations can be caused by several factors. First would be replication errors. When DNA polymerase doesn't catch mistakes and doesn't fix those during replication in S phase, you can get a gene mutation occur. Okay. Another thing would be mutagens. Mutagens are agents in the environment that can cause changes in DNA or mutations in DNA. They can either speed up replication or break DNA strands. Some are naturally occurring. For example, UV rays from the sun. Okay, and some are man-made, like industrial chemicals, specifically some of the heavy metals that are used in manufacturing can cause some pretty severe mutations.